Hello everyone, welcome back to our lectures. Today we'll be talking about protein structure and denaturation. And if you look at what I have there, I have a picture of the green, we call this the green fluorescent protein. Green fluorescent protein. This is the picture of the green fluorescent protein. It has a mix of both this is what we call the beta sheet, the beta-pleated sheet, and then this is the alpha-helix. I'm going to tell you what those are by the time, but this is an example of a protein that has a secondary structure. So that is just an idea of a little bit of what we're going to see the moment we start this lecture. Let's immediately, as usual, let's take it off from there. I have a beautiful quote this time from Reverend or Pastor Sam Adeyemi. Sam Adeyemi is a Nigerian preacher. He is the senior pastor in Daystar Ministries and a motivational speaker. When I was younger, I used to listen to a lot of his motivational quotes, and that actually inspired me a lot. That is why I decided to bring this out to share with everyone. He says, successful people are the failures that refuse to quit, while failures are successful people that quit easily. Reverend Sam Adeyemi. Okay, so I'm not going to say much about this. This will be like a food for thought. Let's immediately get into the class. We have two main learning objectives that we're going to be covering in this class. The first one is that we're going to identify and describe the 4 levels of protein structure. What are those 4 levels? The primary structure, the secondary structure, the tertiary, and the quaternary structure. And finally, we'll describe the denaturation of protein structure. Those are the 2 objectives we're going to be picking out at the end of this class. Let's immediately take it off from there. Primary structure of protein. The primary structure of protein is the linear amino acid sequence in the polypeptide chain. The normal linear amino acid sequence that are held together by polypeptide bond. Remember, the amino acid sequence goes from the N terminal and runs through the C terminal. And of course, linked together by peptide bond. And peptide bond is a type of covalent bond. So the type of bonding that stabilize the secondary structure of that stabilize the primary structure of protein is the peptide bond, or you say, which is a covalent bond. So if these my small red circles represent amino acid, each amino acid that connected, of course, it runs from the N terminal to the C terminal amino acid. And then this bond connecting them, of course, is the peptide bond. And a peptide bond, of course, is an example of a covalent bond. Now, what is the importance of the primary structure? Primary structure determines the secondary and the tertiary structure, and even the quaternary structure of protein. So what it means is that this structure determines the 3-dimensional structure of protein. And for a protein to function, it needs to stay in its native 3-dimensional form for it to become an active protein. Now, variations in the primary structure, this means primary, remember that? Variations in the primary structure of protein usually have significant effect in its 3-dimensional structure and hence the function of that protein. Now, if a protein loses, or if a protein does not have the normal sequence of amino acid it's supposed to have in its primary structure, that is going to affect the overall 3-dimensional structure and hence will affect the function of that protein. A good example is what we see in the sickle-cell anemia. What is sickle-cell anemia? In sickle-cell anemia, somebody who is suffering from sickle-cell anemia have what we call the sickle hemoglobin. The sickle hemoglobin, of course, hemoglobin is the protein that helps in the transport of oxygen in the blood. Now, what happens to hemoglobin is constituted of 2 alpha chains. It has 2 alpha chains and 2 beta chains. Now, the problem lies in the 2 beta chain. Look at what is happening here. Now, the hemoglobin, one of the amino acid is in the beta chain, has been replaced with valine instead of normal glutamate. So the normal beta chain of somebody who is healthy will have the glutamate or glutamic acid in it, whereas someone who is suffering from sickle cell anemia will have that glutamate replaced with valine. And because of this single 1 amino acid substitution in this primary structure of one of the beta units of hemoglobin, it results to the sickle cell anemia, whereby an individual will always have a problem in the transport of oxygen. That is the consequence of a single replacement of amino acid in the primary structure of protein. We'll now go to the next one. We now see structures to do this. I remember the level we talked about peptide was when I showed you this picture before, earlier. In peptide, I said it, the amino acid is written or drawn or is coded from the N terminal to the what? To the C terminal. It is coded from the N terminal to the C terminal. And of course, held together by the peptide bonds. Look at the first peptide bond forms by this carboxylic group here, and then the amino group starting from the next one. So this is the peptide bond, this is the peptide bond. And again, like I said, this is the direction of the peptide bond formation. That is how the primary structure of these proteins are built. So I have a good example here. Insulin. Insulin has two chains, the alpha chain and the beta chain. You see, the alpha chain runs this way. This is the alpha chain. Runs from glycine, which is, of course, is going to be the N terminal, to what? To asparagine, which is what? The C terminal. And then the beta, look at the beta chain. The beta chain runs from phenyl to threonine. Now, and look at what happens here. They are held together. The cysteine of this one and cysteine of this one holds this one together. This one holds cysteine here and cysteine here holds this together. Now, listen, it's important I say this. Now, the primary structure does not involve this cysteine disulfide bonds. No, it is only the linear. This linear sequence is what is the linear sequence held together, like I said, by peptide bond is what is the primary structure of this protein. Again, the long linear sequence of this. So this one is a sequence. This one is another polypeptide chain. So this polypeptide chain that runs from glycine to asparagine is a single polypeptide chain that is held together by peptide bond. And that linear chain demonstrates the primary structure of protein. The same thing happens with the blue ones here. This linear chain is held together individually between one amino acid to other by the peptide chain. And that illustrates again the primary. So this linear sequence illustrates again the primary structure of protein in the beta chain. Let's now go to the secondary structure. Now, the secondary structure of protein is just a regular folding pattern in a single polypeptide chain. Now, remember, in all these structures, we're not talking of two polypeptide chains. We're just talking of only one polypeptide chain here. So, a regular folding pattern in a single polypeptide chain that is stabilized by hydrogen bonding in the backbone. Now, like I said, the primary structure is stabilized by covalent bond or the peptide bond, whereas the secondary structure is stabilized by what? Hydrogen bond. Between what and what? Between the amide nitrogen and the carbonyl oxygen of the peptide of the single peptide bonds in question. Now, it could be the peptide bond can run this way. If it runs this way, this is a single peptide bond, and now you have a hydrogen bond here, hydrogen bond, hydrogen bond, hydrogen bond, hydrogen bond, hydrogen bond, hydrogen, but in a single polypeptide chain. Or it can run this way. If it runs like helically, we call it the alpha-helix, whereby polypeptide chains are held together. The adjacent turn of the polypeptide chains are held together by hydrogen bond. Now, again, it's important to state this. It does not involve the amino acid side chain, no, at all. And there are two types that are recognized: the alpha-helix and the beta-pleated sheet. Now, let me show you this. So, take this as this is a peptide. The peptide bond. This is the continuation of that peptide bond. Let's assume that they were folded at this point. They were just folded. It was a round thread folded again. We're talking about a single peptide chain. Look at what happens. This is the carbonyl oxygen from the carboxylic acid group, and this is the amino hydrogen from the amino group. So both of them are held together. This hydrogen bond in between those in that same chain is what actually stabilizes the secondary structure of protein. And like I said, we have two types: the alpha helix and the beta-pleated sheet. Let's see what it is. Now, in the alpha helix cast structure, a portion of the single polypeptide chain coils in a helical structure. It coils as a helix, like a helical spring. So it coils this way. When you say, oh, that helix doesn't look good, let me put it this way. Yeah, just like a spring. And they're held together by the intramolecular hydrogen bond between the carbonyl oxygen and the amide hydrogen in the adjacent turns of the helical chain. Of course, protein secondary structures are represented in two. There are many other ways you represent them, but because of this class, I'm going to be putting you through just two of them. But just I want you to know about them is just to recognize them. So the protein structures are illustrated in two ways. One, we call it, the first one is the ribbon arrow structure, or the cylinder and plate diagrams. You know, you can say structural diagram, it doesn't matter. For the alpha helix is shown or depicted as a coiled ribbon in the ribbon arrow diagram. So in ribbon arrow diagram, we represent the alpha helix as a coiled ribbon, whereas in the cylinder and plate diagram, it is represented as a cylinder. So just note that as a coiled ribbon at this point and as a cylinder. And of course, we can find a mix of both the alpha-helix and beta-pleated sheet in different proteins. So, but common examples of where we can see them include the keratin. You find them in the keratin in the hair, myosin in the muscle, epidermis in the skin, fibrin in the blood clot, and so many other type of protein examples as we're going to see as we move on. So let's look at this. Now, so this is the typical alpha-helix. Now, this alpha-helix, you see, looks like a helical spring that held together by this intramolecular hydrogen bonding between the NH and the CO of the same polypeptide chain. So now listen. So this is the hydrogen bond. This is the hydrogen bond that holds it into this shape. So this is another hydrogen bond, this is another hydrogen bond, this is another hydrogen bond in a single polypeptide chain. So let's see some proteins that have this type of structures. A good example is myoglobin. You see, myoglobin has the what? Look at it, the coiled ribbon. The coiled ribbon here is showing us what the alpha helix. So look at the coiled ribbon. It has there. So this is the coiled ribbon. This is the myoglobin. The myoglobin, this is the what? Like I said, this is the ribbon arrow diagram of the myoglobin. Whereas this is what? The cylinder and plate diagram of myoglobin. Remember, in the cylinder and plate, the alpha helix is represented with what? With a cylinder. This is a cylinder. Remember that. This is a cylinder. And this is a coiled ribbon. This is a coiled ribbon showing you the alpha. So these are all alpha helixes. This is alpha helix. These are all alpha helix. This cylinder, remember, it is the alpha helix. This is the alpha helical structure. So what I want you to do to be able to recognize this structure when I show you some examples of proteins in your assignment. So let's now go to the next one, the beta-pleated sheet. In a beta-pleated sheet, the sections of a single polypeptide chain fold in a beta-pleated pattern or sheet. It folds like a sheet. Of course, in the ribbon arrow diagram, the beta sheet is depicted by what? A wide arrow. Look at it. It is depicted by a wide arrow. And the wide arrow runs from what? The N terminal towards the C terminal. So you start from the N terminal, goes to the what? To the C terminal. That's how it runs, to the C. Again, it runs from the N terminal to the C terminal. Here it's going to run again from the N terminal to the C terminal. That is how it runs. And like I said, it's like a sheet. It's like just where you put sheet between two wood as a sheet and use it to make like a sheet like something, you know, just like a blind. Now, this is the intramolecular hydrogen bond holding or stabilizing it together. That is the hydrogen bond holding or stabilizing it together. Like I said again, it is represented by what? Plate. In the what? In this cylinder and plate diagrams. It is represented by plate in the cylinder and plate diagrams. And we're going to see that example in the next page. Now, it is stabilized by hydrogen bonds. And of course, most proteins will have a mix of both the alpha helix and the beta-pleated sheet in its native structure. So this is another good example here. This protein is a beta barrel. The beta barrel has an extensive beta-pleated sheet in it. That's what we call it, the beta barrel. If you look at it, just an extensive structure of the beta barrel running around this particular structure. So let's see some other practical example. There are two examples I'm going to show you here. The superoxide dismutase. The superoxide dismutase, the ribbon, this is the ribbon IO diagram. Look at it, running to the N from N terminal to C diagram. So it has an extensive beta-pleated sheet in it for the superoxide dismutase. Remember, the zinc and copper here constitute the prosthetic group, which forms part of the native structure of the enzyme. It's an enzyme, the superoxide dismutase. Now, another good example here is the one I showed you when I was starting this class, the green fluorescent protein, GFP. This is the cylinder and plate diagram. In the cylinder and plate diagrams, look at what is happening there. It actually does have the what? The alpha. Do you remember this is the alpha-helix? But this is the, see, it's represented like a plate in the cylinder and plate diagram. The alpha, the beta-pleated sheet is represented by what? The plate. So this is the plate, and this plate, of course, is showing you, is describing the what? Describing the beta sheet, beta-pleated sheet. This is the beta sheet in the plate. So the green fluorescent protein has a mix of both the alpha-helix and the beta-pleated sheet in itself. Now we'll now start talking about the tertiary structure of protein. You see, the tertiary structure of protein describes the three-dimensional shape of protein resulting from the interactions of the side chain amino acid between themselves, or the side chain of the amino acid with the environment. Now, this doesn't occur in every protein. It has to be with proteins that have particularly prosthetic groups. But these are the two things. So here, now remember, in the first ones we talked about, the primary structure is stabilized by covalent bond or the peptide bond. The secondary structure is stabilized by hydrogen bond. But these ones are, both of these two early structures we talked about, both the primary and the secondary, do not involve the side chains. In the tertiary structure, it is just the side chains that run the show. So the side chains is what makes these interactions. And there are 5 interactions that have been recognized here. The disulfide bridges. Disulfide bridges just involves interaction of a covalent bond between 2 cysteine residues, forming the covalent. Of course, the disulfide bridge is what? A covalent bond. We've talked about this a lot in this class. And then the salt bridge. The salt bridge is an ionic bond that forms between ionized side chains of both acidic and amino acid chain. Remember, the acidic side chain usually have the side chain this. So both of these can also attract each other. Remember, this is charge minus, this is plus for a basic amino acid. So they can easily form a bond. They can form a type of ionic bond between them. That is what we call it, ionic bond or the salt bridge. Then hydrogen bond also can form between 2 polar side chains. Remember, there are a lot of polar amino acids. Like if you have a polar amino acid like serine, ends with, okay, serine ends with OH, whereas you can have another serine that has another OH here. So, wait, okay, let me make it this way. Remember your hydrogen bondings. If we decide to put this one like this, and you decide to put this one this way, you see there will be interaction between these to form this hydrogen bond between the polar side chain. Remember, this particular hydrogen bond is different from what you have in the secondary structure of proteins. Then, hydrophobic interaction is the weak interaction between non-polar side chains as well. So the hydrophobic or the London dispersion forces. Now, lastly, the last interaction is what we call the metal-ion coordination, whereby the 2 side chains that have the same charge are linked via a metal ion. That's what we call it, metal ion. Metal ion coordinates 2 side chains that are charged, that have the same charge. So, example, the 2 glutamic acid chain is this. They could be attracted by a magnesium ion in between them. So that's what we say. So you can have this guy, both of them will be attracted by, like, say, magnesium 2+, and then you're going to have this again. This is what we call this is what we call the metal ion coordination. Metal ion coordination. So in aqueous environment, the interaction of hydrophilic and hydrophobic side chain with water is what determines the overall structure of the protein or enzyme. In fact, particularly enzymes, look at what happens. Enzymes particularly fold in such a way that the hydrophobic side chains are outside in contact with the aqueous environment, while the hydrophobic side chains are buried inside to avoid water. This looks like what we have what? Remember that? In the micelle. This looks like what we have in the micelle. So, and in fact, it is this particular type of mycelic structure that is thermodynamically stable. You know, so if you have a protein that is folded on itself and folded on folded, what it simply means is that if I use a different color here, let's say blue, the amino acid side chains that are polar will be segregated outside so that they can make contact with the aqueous cellular environment. Whereas So if I label that guy now, assuming I want to label it now, this will be the polar ones, the polar side chains. Whereas inside here is where we have the non-polar ones. And this is typical of what the shape of most enzymes that gives them that globular structure for them to actually remain in their native state and carry out their what? Biological functions. So I have a very good diagram illustrating all those levels of protein structure here. So let's look at it. All those levels of, so I have this is my polypeptide chain running this way. Look at it. It started here, runs this way, runs this way. So this is my polypeptide backbone. Let's start going down from here. In here now, you see, first of all, this is a disulfide bridge. In this disulfide bridge, two cysteine residues comes together to get themselves interact with this disulfide. Remember, this is the disulfide, and this disulfide I told you is also a covalent bond. Remember, that's why when I showed you the picture of insulin, I told you that the disulfide bond is not part of the primary structure. It is part of what? The tertiary structure of proteins. Then let's go to the next one. Now, after the disulfide bridge, what do we have here? We have two serine. Remember, serine is a polar amino acid that has the OH group as the functional group. The OH group is polar. So look at two serine residues attract each other to form this hydrogen bond. This is the second one. So let me label, I can label this one as the first one. Let me say first. This is the second interaction. 2, the hydrogen bond between two polar side chains. Then 3, metal-ion coordination. Remember, aspartate or aspartic acid is an acidic amino acid that has a negative charge. 2 of those negative charge is stabilized by magnesium ion in between them. That's what we call this the metal-ion coordination, because this metal ion is coordinating these two same charges to interact with each other. That was what we say is a metal-ion coordination. This is number 3. And then going to number 4, we see the non-polar side chains. When the non-polar side chains are close to each other, they tend to interact. That particular interaction is called hydrophobic or dispersion, or you can say London dispersion forces. So this is what? The 4th one. And the last but not the least is salt bridge or the ionic bonds. How does the salt bridge or ionic bonds form? It forms between two oppositively charged groups. So here, lysine is a basic amino acid that has what? The ammonium ion here as the charge group, which is charge +1. Whereas aspartic acid or aspartate has what? The carboxylic group as the ionizable group. What happens here? Both of these two guys come together to attract each other to form the ionic bond or the salt bridge. This is the 5th one. So your main duty will be to recognize this for me in exam. So I can draw this type of structure and ask you to label or name the various types of tertiary structure interactions of a particular protein. When I go to the next thing, the final one, the quaternary structure of protein. Now, before we continue, the quaternary structure of protein is almost the same with the tertiary structure of protein. The only difference is that in quaternary structure of protein, a protein will not involve more than one polypeptide chain. However, the same type of interactions in the tertiary structure, like we just listed, will also be the type of interactions that will take place in the quaternary structure of protein. So the quaternary structure of protein is the 3-dimensional structure of proteins with more than one subunit. More than one sub means more than one subunit, or you can say, or more than one polypeptide chain or backbone. Now, it forms larger or more compressed proteins. So let's define subunit. A subunit is a polypeptide chain that has a primary, secondary, and tertiary structures in it. And of course, this is particularly seen in most conjugated or complex proteins. In most conjugated or complex proteins, remember we said they contain, in addition to the polypeptide chain, they also contain other non-protein substances. And we call those the prosthetic groups. So the conjugated proteins contain, the conjugated or complex proteins with quaternary structures contain subunits and the prosthetic groups. And then this prosthetic group may be, the prosthetic groups may be organic or inorganic. Example, heme is the prosthetic group that is found in hemoglobin and myoglobin. And they help with the transport of oxygen. That is why both hemoglobin and myoglobin are very efficient transporters of oxygen, both in the muscle. Myoglobin is in the muscle, hemoglobin is in the blood. And I have a picture of a heme. This is a picture of a heme I don't expect you to memorize this. What I only expect you to do is just to recognize the structure of the hem. This is the structure of the hem. So I don't want to bother you with a lot of terminology. Just know that this hem, this is the hem structure, and the hem helps myoglobin, Mg, and hemoglobin in transport of oxygen. That's just what you need to know. And you don't need to bother about what the structure. But like you see, this is not a protein. This is the non-protein part. And it also has a bridge of what? Ion. That it is this ion too that helps it when it oxidizes between ion 2 to ion 3 and ion 3 back to ion 2 is this process that helps with the transport of oxygen in blood and muscle respectively. We now finish this quaternary structure by taking a look at hemoglobin itself. So this is hemoglobin. Hemoglobin have 4 subunits. That means it is very big. It has 4 polypeptide chains. Those 4 polypeptide chains include the 2 identical alpha chains and 2 identical beta chains that are held together by hydrophobic interactions. Now, it contains 4 heme groups. Each heme group for 1 subunit. Look at it. So this is the first subunit. Let's see. I try to use a structure that shows you the different ones. So now this is the alpha 1, the first alpha unit. They're identical. This is alpha 1. This is alpha 2. Now this is beta 1. This is beta 2. Those are the 4. So you see, these are the 4 polypeptide chains of 4 subunits that make up what? The hemoglobin. And each of them have the prosthetic. This is the heme. This is the heme prosthetic group that helps you to do that. So each of them have this is the prosthetic group of this one, this is the heme of this one, and this is the heme of this one. And it's important to know that hemoglobin has all the 4 levels of protein structure, which include the primary, of course. Remember, it has the primary structure, which is the sequence. Every protein must have a primary structure, which is the sequence of amino acid. It has the secondary structure. If you look here, you see the coils, the alpha helical coils, those helix. You see, this is an alpha helix. This is the alpha helix. This alpha helical coil represents one of those secondary structures. And then the tertiary structure means it is folded on each other, and there are interactions of particularly the hydrophobic interactions holding them together. So it has the tertiary, and finally, because it has more than 1 subunit, it has 4 subunits, it has the quaternary structure. It has the quaternary structure. So it has all the 4 levels of protein structure represented in the hemoglobin. Now let's talk about the protein hydrolysis. Protein hydrolysis, we've talked about this before, is just the breaking down of the amide bonds or the covalent bonds and releasing individual amino acid. So it can happen in different ways. Heating of a peptide or a protein in the presence of an acid or a base will cause it to break into smaller peptide or amino acid. It can also be done by digestion, but in the intestines. So digestion of food or proteins in the intestine also involves hydrolysis. So, and they are catalyzed, of course, by proteolytic enzymes, or we call it the protease enzymes or the peptidases. Remember when I told you, when I showed you how amides or rather peptides are formed, I showed you this 4th reaction. The 4th reaction is the formation of peptides, which is the process of amidation. But that's not what I'm looking. I'll be looking at the opposite reaction. If you look at the opposite reaction now, this is a dipeptide. Of course, there are 2 of them. This is a dipeptide. 2 amino acids, R1 and R2 here, telling you that 2 amino acids. This is a peptide bond. Can be broken together by hydrolysis. This hydrolysis, like I said, it could be it could be acid. It will give you the hydrogen plus. It could be a base, or it could be an enzyme in the digestive system. We'll break this up. What they do is to come to chop this apart, and then using water molecule. Remember, it's hydrolysis. You're using water molecule. So one OH group will now be attached here to form. One of the OH group will now be attached here to make this completely to form this carboxylic group, and the other one is going to accept the hydrogen remaining from water to be attached here. So remember, you are using water at this point, so you can just say, oh, look at our water here. Okay, I'm almost looking for it. Yeah, remember, water is H plus and OH minus. Remember that. Okay, that doesn't fit well. I want to make sure I don't go beyond the boundaries. Okay, this is H plus OH minus are the two things that you add and use it to break down these peptide bonds into the individual amino acid. That is the process of protein hydrolysis. So you must at least be able to do what? Show me the different product of a peptide from their individual amino acid in exam. All right. When I go to the last thing we need to be talking about, the last objective, which is protein denaturation. Protein denaturation is a process that disrupts the characteristic native structure, which are the secondary, the tertiary, and the quaternary structure of protein. It results to the loss of biological functions by a protein. Now, what is this native structure so-called? I've used it a couple of ways. A native structure is the three-dimensional, the natural three-dimensional conformation of a functional protein with interactions of the secondary, the tertiary, and the quaternary structure intact. And what simply means is that in the native state, the protein has all the three-dimensional structure and retains its biological functions. And of course, when does denaturation occurs? It occurs when a protein is exposed to harsh physical or chemical environmental condition and may cause some protein to even precipitate and become permanently damaged. This is a good example of a protein. So this protein, I made it up myself. This is a protein that is supercoiled. When you denaturate it by those conditions, those harsh conditions, what happens? It unwinds it completely and releases it, and so that it loses the secondary. Denaturation does not affect the primary structure. It only affects the secondary, the secondary, tertiary, and the quaternary structures. So look at here. It has just unwound this protein to become a single linear polypeptide chain. At this place, it has lost its biological function. And of course, it lost its three-dimensional structure. This is the 3D here. It has lost it here. And in addition to losing the 3D structure, it has also lost its biological functions. Okay, that is denaturation. So what are the factors, the conditions that cause denaturation? They include the following: heat or ultraviolet light or mechanical agitation can break hydrogen bonds, ionic bonds, and hydrophobic bonds. Detergent or organic solvent, corrosive detergent or organic solvent can disrupt hydrogen bonds, ionic bonds, and hydrogen and hydrophobic interactions. pH, or you can say strong acid or strong base, very concentrated acid or base can disrupt the hydrogen bonding, ionic bonding, and may lead to hydrolysis of the protein. And once you hydrolyze them, what happens? They will not be effective anymore. Then the heavy metals. Heavy metals can also disrupt hydrogen, can also do what? Disrupt the three-dimensional structure of protein. And what they usually do is that they form irreversible complexes with the thiol group of cysteine. They form permanent structures that cannot be undone, and in fact result to the precipitation of proteins as insoluble heavy metal complexes. And this is the basis for what? The heavy metal poisoning. And we talked about a little bit of this when we talked about the formation of disulfide bonds. So this is the basics of actually heavy metal poisoning. Like, and heavy metals here include lead, mercury, and even silver. And with that, we have almost come to the end of this lecture. But before we go, please, I know this is not a clickable. In this particular video, you cannot click this link to watch them. But go back to your note, the note in PDF, click this and watch these particular protein videos, protein and denaturation videos. They are very, very important. They are very interesting videos that could also offer you more insight to both to the structure and functions of proteins. And having said that, we've come to the end of this lecture. Thank you once again for listening. Bye at this point.